This video is about chemotaxonomy, serotaxonomy, and cytotaxonomy. Taxonomy is the practice of identifying different organisms, classifying them into categories, and naming them. All organisms, both living and extinct, are classified into distinct groups with other similar organisms and given a scientific name. First, we are going to learn about chemotaxonomy. All organisms coexist in ecosystems and interact with each other in various ways in which chemistry plays a major role. Many approaches are there towards taxonomy of organisms. Chemotaxonomy is the modern approach, especially for plants now. The chemical compounds studied most are proteins, amino acids, nucleic acid, peptides, etc. Chemotaxonomy is the classification of plants and animals based on similarities and differences in biochemical composition. Chemotaxonomy is the classification of plants and animals based on similarities and differences in biochemical composition. Merriam-Webster defines chemotaxonomy as the method of biological classification based on similarities and dissimilarity in the structure of certain compounds among the organisms being classified. Some argue that as proteins are more closely controlled by genes and less subjected to natural selection than the anatomical features, they are more reliable indicators of genetic relationships. Physiology is the study of working of organs in a living being. Since working of the organs involves chemicals of the body, so these compounds can be taken as biochemical evidence. The study of morphological change has shown that there are changes in the structure of animal which result in evolution. When changes take place in the structure of a living organism, they will naturally be accompanied by changes in the physiological or biochemical process. John Griffith Vaughan was one of the pioneers of chemotaxonomy. Chemotaxonomy, also called chemosystematics. In a nutshell, chemotaxonomy is the biological classification of plants and animals based on similarities and differences in biochemical composition. Next, chemotaxonomy classification. The phenolics, alkaloids, terpenoids, and non-protein amino acids are the four important and widely exploited groups of compounds utilized for chemotaxonomy classification. The system of chemotaxonomy classification relies on the chemical similarity of taxa. Three broad categories of compounds are used in plant chemotaxonomy: primary metabolites, secondary metabolites, and semantics. So primary metabolites are compounds that are involved in the fundamental metabolic pathways which are utilized by the plant itself for growth and development. For example, citric acid used in Krebs cycle. Then secondary metabolites are the compounds that usually perform non-essential functions in the plants. They are used for protection and defense against predators and pathogens and perform non-viral infections and non-vital functions. For example, alkaloids, phenols, glucosinolates, amino acids, etc. But primary metabolites are compounds that are involved in the fundamental metabolic pathways which are utilized by the plant itself for growth and development. For example, citric acid used in Krebs cycle. Then semantics, information carrying molecules like DNA, RNA, proteins. Chemotaxonomy in animals. The body of any animal in the animal kingdom is made up of a number of chemicals. Of this, only a few biochemical products have been taken into consideration to derive evidence for evolution. So protoplasm, every living cell from a bacterium to an elephant, from grasses to the blue whale has protoplasm. Though the complexity and constituents of the protoplasm increase from lower to higher living organisms, the basic compound is always the protoplasm. Therefore, all living things have a common origin point or a common ancestor which in turn had protoplasm. Then nucleic acids, DNA and RNA, are the two types of nucleic acids present in all living organisms. The structure of these acids has been found to be similar in all animals. It has been proved in the laboratory that a single strand of DNA of one species can match with that of the other species. If the alleles of the strands of any two species are close, then it can be concluded that these two species are more closely related. Digestive enzymes are chemical compounds that help in digestion. Proteins are always digested by a particular type of enzyme like pepsin, trypsinogen, etc. The complexity in the composition of these enzymes increase from lower to higher organisms but are fundamentally the same. Protoplasm, nucleic acid, and digestive enzymes. Importance of chemotaxonomy. Chemotaxonomy has been used in all levels of classification. Chemical evidences has been in all the groups of plant kingdom, starting from complete organization complete organisms like fungi and bacteria up to the most highly advanced and specialized group of angiosperms. Chemotaxonomy-based selection is a prerequisite for the successful natural product research. The rise of chemotaxonomy is mainly due to the advancement in analytical technique for chemical analysis and can detect even trace amounts of chemical compounds. Then serotaxonomy, next topic. Serotaxonomy is defined as the portion of biology which is concerned with the nature and interaction of antigenic material and antibodies. Smith defined it as the study of the origins and properties of antisera. When foreign cells or particles or antigens are introduced into the organism, antibodies are produced in the body. Antisera. Serotaxonomy developed and became popular in Germany, which has been an active center since the beginning of this century. Nuttall was the first biologist to compare the immunochemical specificity of serum proteins for systematic purposes. In other words, serotaxonomy is the classification of very similar plants by means of differences in the proteins they contain. The technique is based on the highly specific relationship between antigens and antibodies produced in response to them. Methods used in serotaxonomy. In this method, a crude protein extract of a particular plant taxa or antigen is injected into the bloodstream of an experimental animal, usually a rabbit or a rat, to develop antibodies. In response to the specific antigen injected, a specific antibody is produced in the blood of the animal. The serum, termed the antiserum containing the antibody, is then collected and made to react in vitro with the antigenic protein as well as protein from other related taxa of which the affinities are in question. Serological reactions between antibodies and antigenic material results in the formation of a precipitate that is called precipitin reaction. Then Cross in 1897 showed that this reaction indicates similarity of antigens. The degree of protein homology is determined by the amount of precipitation and hence it is taken as a phylogenetic marker and taxonomic character. However, according to Moritz and Jensen in 1961, crude protein extracts should not be used as antigens and has been the normal practice of serotaxonomic work. This is because crude protein extracts contain a large number of proteins which stimulate the production of a vast range of antibodies which differ in their specificity and reactivity. Some are produced in abundance while others are hardly detectable. Several serological methods are now available. Immunodiffusion in agarose gel, rocket immunoelectrophoresis, enzyme-linked immunosorbent assay or ELISA. Role of serotaxonomy. Serological studies using crude plant protein extracts have been widely used in elucidating the taxonomy of wide variety of higher level taxa and in estimating phylogenetic relationships. A close relationship among the Magnolidae, Hamalinidae, and Comiflorae of the angiosperms has been found based on comparative serological studies of their major seed proteins. This has refunded the idea of their independent evolution. Then serological studies have shown that the genus Lenodendron is quite distinct from the other members of the family Magnolinus, and genera Magnolia and Michaelia are most closely related within the family. Next is about cytotaxonomy. Cytotaxonomy is a branch of taxonomy that uses the characteristics of cellular structures to classify organisms. In cytotaxonomy, the chromosomal configuration of an organism is the most widely used parameter to infer the relationship between the two organisms. The inference of species relationships is based on the assumption that closely related species share similar characteristics in their chromosomal setup, referred to as karyotype. By analyzing the similarities and differences in chromosomes, karyotype evolution and species evolution can be reconstructed. The number, structure, and behavior of chromosomes is of great value in taxonomy, with chromosome number being the most widely used in quoted behavior. Chromosomes, quoted character. Chromosome numbers are usually determined at the metaphase stage during mitosis. Metaphase stage during mitosis, usually the diploid chromosome number is referenced unless dealing with a polyploid series, in which case the base number or number of chromosomes in the genome of the original haploid is quoted. Another useful taxonomic character is the position of the centromere. Meiotic behavior may show the heterozygosity of inversions. This may be constant for a taxon, offering further taxonomic evidence. Cytology has contributed to tracking the evolutionary history of many organisms, especially primates and flowering plants. Then cytotaxonomic approach in classification. Genetic complement. DNA is the essential material of heredity. It comprises the genome and plasma. It is believed that the heterochromatic segments are associated with differences in the metaphase thickness. Even now, it is known that a given amount of DNA and protein is stimulated at mitosis to become distributed into a particular member of chromosomes. Then DNA hybridization. The hybridization between single-stranded DNA components from different origins can occurs provided psychochemical means for assuming genetic relatedness among the species. It is known as DNA is extracted from organisms and made to hybridize in vitro with cell lines of other organisms. The DNA matching technique is much easier to solve for solving complex taxonomic problems. Then karyological studies. Chromosomal cytology has been more extensively used by plant taxonomists rather than animal taxonomists. The karyotype, characterized by chromosome number, size, and morphology, is a definite and constant character of each individual species. The number shape banding of chromosomal can be determined by using various dissecting and staining technique. Chromosomal taxonomy can be quite useful both in determining the phylogenetic relationships of the taxa as well as the segregation of sibling cryptic species. Then significance of cytotaxonomy. The role of cytotaxonomy is very important in taxonomic studies. Cytotaxonomy is more significant over physiological taxonomy because this process is dealing with the comparative study of chromosome, and with this method minute variation among the individuals can be detected. DNA is present in every chromosome, and the variation in each DNA is responsible for variation among the individual species, genus, and everything. So cytotaxonomy is a part of taxonomic biology that deals with the classification of organisms based on their function and cellular DNA structure. So thank you for listening.